Good morning and welcome to the Marcus Wallenberg Prize Symposium. So last night we had a really tremendous award ceremony. For those of you who were there, we hope you enjoyed it very much. Today we're going to talk about the science and the application, and I'm looking forward to this very much. So it's great to see so many people here in the room. We have great diversity in terms of people from different parts of the world, but we also have great diversity in sense of young researchers. We have academics in universities and a lot of industrial interest as well. I think the large number of people here and also online, we have very many people online as well watching, shows great interest in this area of lignin and the applications of lignin that are being developed. So in a moment we'll have some of the talks, but first I would like to ask Marcus Wallenberg to say a few words.
Great to see you all here. This is not all about partying. Perhaps this microphone is not what it should be. Anyway, we're here actually to dig into the secrets of lignin. And as we heard last night, those secrets seems to be more than one. So I'm really happy about this. It also appears that we are coming closer and closer to perhaps what is the real discussion for forest products and forest industry in the future on what are we going to do with the tree fiber going forward. It's obvious that the last 20-25 years from an industrial point of view has changed the landscape completely. And the big question is, how are we going to take scientific breakthroughs such as the work around lignin, how do we take that into the next phase on how to apply that from an industrial point of view going forward? I think that is really, really important. We're so happy about having the laureates here to help us out a little bit more today. I see that they have split themselves completely. One sitting up there and one sitting down there. I'm not 100% sure what significance that has. Maybe something happened last night in the bar that we're not really 100% about. And I just want to reiterate the question about young researchers. I should tell you that perhaps one of the biggest satisfactions of having been involved with the prize for quite some time now is actually that we have more young researchers around us and who are showing great progress, great interest in bringing this forward. So I'm extremely happy and thankful for that your interest and that you are here with us today. So I wish you all a good day. I hope we're going to have a really interesting session. Thank you.
So the symposium is divided into different sections. First, we're going to have a scientific session where we will hear about lignin and we will hear from the laureates. And then after some awards to young researchers, we will have second and third sessions on the industrial applications. Okay, so first we're going to hear from Claire Halpin. And we're very honored that Claire has agreed to give us an introduction to lignin. Claire has been working on the biosynthesis of lignin for many years. She works at the University of Dundee, and she's focused particularly on lignin in grasses. And she's made quite some considerable advances herself. So it's a great delight, Claire, to invite you to come up now and give us a talk that introduces the work of our laureates this year.
Well, thank you, Paul. I feel the honour is all mine to be asked to introduce this wonderful symposium. And I hope by the end of it, you'll be convinced that we can now make anything from lignin, including lots of money. But I'm particularly here to give you a perspective on the work of our laureates. And in order to do that, it inevitably means giving you something of a historical perspective. But I want to go just a little bit further back than the 30 years or so that they've been working in the field. 400 million years further back, in fact, to the time depicted in this artist's impression of an early Devonian landscape, when plants were just beginning to come out of the water where they had existed up until then and to colonize the land. And in order to do that, they needed to evolve many adaptations, some of which you can see on this cross-section of an exquisitely preserved fossil of this small little plant, Rhynia. And in this cross-section of a stem, you can see that this plant had begun to develop dense chemical material in its outer layers and also in the center, where it was beginning to develop a vasculature, the beginnings of xylem. And we now know that these dense chemicals included some of the precursors of lignin. Of course, Rhynia could only grow to be about 10 centimeters tall, so further adaptations and evolution was needed before trees could evolve. Those chemicals needed to evolve into polymeric lignin to provide UV protection, to waterproof and rigidify the xylem so the trees could conduct water all the way up from the roots to the top of the trees, and to basically make the wall less penetrable, to make it impenetrable to pathogens. And so I wanted to just illustrate this with a picture of a seasonal tree, this holly from my garden. Without lignin, this tree could really only grow as tall as the moss that you can see here growing on the base. But lignin isn't important just for the wood in the stem. Clearly, that's one of its major requirements in plants. But it's also important, for example, in lots of little specialized tissues in plants. For example, the seed coat within the berries will have lignin in there, and that's really important so that when the birds eat the seeds, it can travel through their digestive system and get dispersed when they excrete it into other environments. So just to look in a little bit more detail about what lignin is actually doing in the plant cell wall, you can see here that the major components of plant cell walls are these yellow microfibrils made up of crystalline and semi-crystalline cellulose, which is linear chains of sugars. But in a very mature wood cell wall, that cellulose and the hemicelluloses become completely encrusted in this phenolic polymer lignin that waterproofs and strengthens and glues the whole structure together. And because of that complicated and gluing structure, lignin is often seen as a problem when we want to get to cellulose sugars in industrial processes. For example, it needs to be removed in pulp and making fibers for other purposes in order to get to nice clean cellulose fibers. And also in more modern processes where we might want to break down the cellulose into its component sugars, we can do that by pre-treating to get rid of the lignin, then adding enzymes to break down the cellulose into sugars. This is a process known as saccharification. And those sugars can then be fermented by microbes into all kinds of useful chemicals. But the start of this process is to, first of all, get rid of the lignin. And we do that using pre-treatments of harsh chemicals, pressure, and high temperature. And that lignin in the past has often been burned for energy to help fuel those processes. But as you'll hear in many of the talks later today, there are now many more innovative and novel ways that we can use that lignin to give us higher value products. So I want to turn now to giving you a little bit of a perspective on some of the wonderful work by our laureates. So I'm going to really talk in the next couple of slides about what we knew 30 years ago or so when they started in the field and what we now know. So 30 years ago, we had the idea that lignin was made up of three particular types of units, these three alcohols here that give rise to the H, G, and S units in lignin. We knew that enzymes, peroxidases and lactases could connect those units in many different ways with many different types of chemical bonds. But we didn't really have a very good idea of the structure of what that lignin might look like. And as I said to you, when lignin is removed in industrial processes, the harsh chemicals that we use completely destroy the structure. So getting to understand the structure of native lignin in the plant cell wall has really been a huge advance. And you can see over here one of the recent structures that John Ralph has come up with for the structure of native lignin. This is an area that John's been working in for the past 30 years and has evolved through many different models to get to a much better understanding of what lignin really looks like in plant cells. And you can see within this model these different S and G monomers and all these different linkages that bring lignin together in a very complex and irregular polymer. John's work has also increased our understanding of how these units get connected together within the cell wall. And we now know that it's through combinatorial radical coupling reactions. In other words, any chemical that is able to get out into the cell wall and that can make a free radical can potentially be incorporated into lignin. There is no underlying order or sequence of events that is very directly tailored the way there would be in some of the carbohydrate molecules. So really a sea change in our understanding of lignin structure due to the work of John Ralph. Another place where we've really refined our understanding is on the biochemical pathway inside cells that makes those monomers. Now, it's far too early in the morning for you to take in the detail on this slide. I just want you to look at the colors. So 30 years ago, the pathway, actually, we had known for quite a long time, and some of the enzymes on it. But we used to think that this orange pathway was the dominant route through the pathway. And through the work of Wout, Boerjan and many others, we now know that the dominant route of that pathway in plant cells is in this blue route, and in grasses the blue and the pink routes. So we know a lot more about how the process of making these monomers happens, making these units happens within plants. And there have also been many discoveries, several of which Wout contributed to or was the lead on to find new enzymes and even new components that go into lignin. So the major technical advance that really allowed us to understand the pathway for lignin biosynthesis better was the ability to be able to genetically manipulate lignin in plants. And it's a massive honor to be able to say that in front of Professor Mark van Montague, who was really the pioneer of this whole technology. So using genetic manipulation, for example, I'm just going to give you one little example here. We've been able to switch down this CAD enzyme that is involved in converting aldehyde molecules inside the cell into alcohol molecules, and those alcohol molecules are the direct units that normally go into lignin. What we discovered when we switched down the CAD gene was that actually those aldehydes are capable of being incorporated into lignin themselves. So we had a new pathway and new monomers, and in other work, manipulating other genes on the pathway, we've discovered new enzymes. We've also sometimes had other novel results. For example, when we manipulated that CAD gene, and this is an experiment in tobacco, we found that the wood became red because of these new components in the lignin. And Wout, again, was very, very quick to be able to move this kind of technology over from simple model plants into real trees. This is an example of manipulating that CAD gene in poplar, and you can see again this bright red color. So another complete seed change in our understanding of how lignin is made within plant cells. Wout has also been a great champion of the omics. This is where we've been able to take what we know about all of the genes in particular species, and Wout was involved in the development of the full genome sequence of poplar, looking at all of the transcripts within a cell and looking at all of the metabolites within plant cells. And this allows us an unbiased way of getting to discover new genes and new enzymes and new processes on the lignin biosynthetic pathway, and many discoveries and new understanding has been made in that way. John and Wout together have also pioneered new analytical techniques for looking at oligolignals, several of those monolignals connected together within the cell. And John has really been a champion in the use of 2D NMR to better understand lignin structure. And many, many people now use this technique. And what we now know 30 years on is that there's not just 3 monolignals, like I showed you a few slides back, but more like 30 or more different compounds that can be incorporated and have been discovered, have been found in lignin, both in manipulated plants and quite often in native lignins as well. And before I go on to how we might use that lignin, I also just want to pay tribute to the importance of field trials and to Wout's role in championing the importance of field trials. You heard some of that in his speech last night. On the left here, there's a picture of a field trial of these CAD antisense poplars that was carried out in the UK many years ago, that what was involved in. You can see from the red colour in the lignin that the lignin is modified. And you can see from the picture of the plants up at the top, I hope you can see at least, that the modified trees, the trees with modified lignin, grew absolutely indistinguishably from unmodified trees. We couldn't tell the difference by any of the things that we measured until we took off the bark and looked at the lignin. More recently, Wout has manipulated the same gene, but in a much more effective way, using a hairpin construct that really knocks down the activity of the gene much more severely. And in this case, you can see up here in the top photographs, these plants have grown bent away from the direction of the wind, so they're not able to hold themselves upright in quite the same way as an unmodified tree would. And this just shows you how important it is to always take your plants out into the field and really see how plants will behave in a natural environment. This work really shows the parameters that we have in fine-tuning how we would manipulate lignin in plants and in forest trees so that we can use them for industrial benefit. What would we actually use some of those novel lignins to do? So, for example, we could engineer lignin to be more uniform so that when we do deconstruct it and extract it, there's a higher yield of specific chemicals, which would be more useful to industry. We could engineer the bonding in lignin to make it easier to extract in the first place. And John will talk later about his zip lignin, which I think was also mentioned last night. And as I've shown you some examples, we can actually incorporate completely new monomers, which might give us new opportunities to make novel products. So what kind of products could we make? Well, actually, we can't do much worse than going back to nature. You'll hear later today about how we can make products, coatings from lignin that are waterproof and can protect from UV. You'll talk about boards and composites that could be made that are strong and glued together through lignin, and many products where the use of lignin may make the products have a little bit more longevity because it is relatively recalcitrant to break down in the environment. It does, of course, break down, but much more slowly, and sequesters carbon for longer. And many, many other chemicals and applications that I'm sure we don't even conceive of today. So without further ado, I'm going to hand over to the laureates to give you more in-depth stories of some of the pieces of work that they've done. But I also want to thank the organisers very much for inviting me to come and give this introduction.
Thank you very much, Claire. That was a marvellous overview. So now we're going to hear from the laureates, and we're going to see how their technical skills and their interests have been very complementary to make really big advances in the field. So first we're going to hear from Professor Boerjan, who has been exemplary in the molecular genetics side and biosynthesis of lignin and application into trees and engineering. And then we will hear from Professor John Ralph, who has developed techniques for analysis of lignin and demonstrated great diversity and understanding of the structure of lignin. So first I'd like to ask Professor Boerjan to give his laureate lecture. Thank you.
Good morning, everyone, and thank you very much, Paul, for the introduction. And again, I would like to thank the Marcus Wallenberg Foundation for this prize. It's a true honor to speak about parts of my career. So, So the main aim of the research in my team in Belgium is to support transition from fossil-based in a bio-based economy. And we believe that woody biomass is a very promising resource because many products that are nowadays made from oil can be made from woody biomass. And the reason is that woody biomass, all the fractions of woody biomass can be used in society. There is the fiber that can be converted to pulp for packaging and tissue and all kinds of new innovations. The cellulose and the hemicellulose polysaccharides, they can be depolymerized in a process called saccharification to primary sugars, and these primary sugars can be fermented to, for example, biofuels, but also building blocks for bioplastics, detergents, etc. And the lignin can nowadays also be catalytically fractionated into building blocks for the chemical industry. And lignin, as you already heard from Claire, is a main limiting factor if you want to convert wood into either pulp and either into, in a biorefinery, into primary sugars. So there's quite some interest in reducing the amount of lignin in trees or in plants in general, if they are used to improve digestibility. And this can be, so either reducing lignin content is very important, but you can also change the lignin composition such that the lignin becomes more reactive to the chemicals and is more easily broken down. And this is an area of research where my group has been doing quite some research in. So to recapitulate, this is the monolignol biosynthetic pathway, and lignin is mainly composed of coniferyl and synapyl alcohol and actually minor amounts of paracoumaryl alcohol. Thus, when these units are incorporated into the polymer, they are called H, G, and S units. The pathway has been very well described, and that means that we can overexpress or downregulate the genes in the pathway. And in this way, we can create plants that produce a lower amount of lignin or a different lignin composition. And so when these monomers are translocated over the plasma membrane and arrive in the cell wall, they will be oxidized by laccases and peroxidases into monolignol radicals, which will be combinatorially coupled into these standard lignin polymers which grow between the cellulose microfibers in the cell wall. And they make that cell wall strong and hydrophobic such that plants can grow upward and transport water and solutes through the vascular system. Now, today I would like to give you 3 stories. One is how we moved from gene discovery in the model plant Arabidopsis up to trees grown in the field, trees with improved wood properties. A second example is how we can use gene editing using CRISPR-Cas to tweak enzyme activity. And in a third example, I would like to show you how we can make completely novel lignin polymers with different properties. So the first is about gene discovery. Actually more than a decade ago, we asked ourselves the question, are there any roots in that lignin biosynthetic pathway that are undiscovered? Are there any new genes playing a role potentially in bypass pathways? And that was a curious question in a way, because we were looking for something that we didn't know that would exist. But you can do that in biology because you can make a co-expression analysis. It's a kind of guild by association approach. It actually means that we are looking to genes that have an unknown role, an unknown function, and we investigate, we try to identify such genes that behave exactly the same as all the other genes in the lignin biosynthetic pathway. So without going into more detail, so we make a co-expression analysis, so we make a network of co-expressed genes. And we found that in subnetworks B and C, we found all the known lignin biosynthetic genes plus a few other ones. And so these are, of course, prime candidates to also play a role in lignin biosynthesis. And the way we study the role is by analyzing mutants. So at the left side here, we have the wild type and 2 mutants in the CZ gene. And then we made sections through the plant, through the stem, and we stain them with a lignin stain. And as you can see, the amount of the intensity of the lignin stain is much reduced. And there was a collaboration with Claire Halpin. And of course, because the lignin stain is reduced, we hypothesized that those plants make less lignin. And of course, we have to support that with wet chemistry data. That's what we did. So these plants, they have 30% less lignin. And then in collaboration with John Ralph, we found that the frequency of H units was dramatically increased. In wild-type plants, we only have 0.3% of H units. It's really a minor unit. In the mutant plants, the most abundant unit was the H unit, 44% all of a sudden. So if we knock out that gene, we have a less lignin and a strongly altered lignin composition. But these data do not actually show us where the gene plays a role in the pathway. And to investigate what the gene is actually doing and where it is positioned in the pathway, we use comparative metabolomics. It means that we extract metabolites from wild-type plants and from the mutant, and we separate that on UPLC in one direction based on retention time, and then in a second direction based on their mass over charge ratio. And so we can separate about 3,000 peaks from a single extract. And using mass spectrometry, we can fragment those metabolites, and based on the fragmentation pattern, we can puzzle the compound back together. So this analyzing the fragmentation pattern tells us the structure of the molecule. So if we do a comparative metabolite profiling of the wild type and the mutant, we saw that caffeoyl shikimate was a compound that massively accumulated in the mutants. And this rang a bell because caffeoyl shikimate was an already known pathway intermediate. So to discover the role of the enzyme, we heterologously produced the enzyme, and then we fed it with caffeoyl shikimate as a substrate. And using these enzyme assays, we saw that caffeoyl shikimate is converted into caffeic acid. So we discovered a new path, a new piece of the pathway. But it's an important piece because if you knock out the gene, the production of lignin is much reduced, and because we have 30% less lignin, and at least part is compensated, the flux goes into paracomydyl alcohol, which is a very abundant monomer. Now, very interesting is that if you saccharify the stems of these plants, the saccharification is enormously increased. So here you see a time course experiment where we plotted against the percent of cellulose that is converted to glucose by an enzyme treatment of the biomass. In wild type, only 18% is converted in our mutants, in the knockout mutants. This is increased by 4-fold. Almost all the cellulose got converted, and this without any biomass pretreatment that is usually used to open up the cell wall and destroy the lignin to a certain extent. That was very promising. So therefore, we translated that into poplar. And we achieved poplars, and this is greenhouse data, that have up to a quarter less lignin. And these trees, much to our surprise, did not have any yield penalty. So therefore, we took those trees further into the field. We propagated them. This is collaboration with the Ilvo station, and we flew over a drone. And so we have the wild type and 2 transgenic lines, each in 48 copies, and this divided in 6 blocks in the field, and this is to be able to do proper statistics. So if you look to the drone flight, there does not seem to be any yield penalty. This was a big surprise because I would really have put my hand in the fire that we would have a yield penalty, but there doesn't appear to be one. But of course you have to measure this. And so we harvested 3-year-old trees, did very strong statistics. We didn't have any yield penalty. And lignin measurements told us that there was a reduction in lignin content by 13%. It's less reduced as compared to the greenhouse, but that's something we always see. And that translated to a 90% increase in saccharification efficiency. So it means that upon an alkaline pretreatment, so it means that the biomass is more easily saccharifed because there is less lignin, cellulose microfibrils are more accessible to the enzymes. And together with Troy Runge's lab in Wisconsin in Madison, we performed kraft pulping experiments with the wild type and the 2 transgenic lines in 8 different conditions with ranging active alkali, sulfidity, and H factor. And then based on these data, and these are actually quite fresh data, models can be built and then you can ask questions to the models. So these data show you, or the model tells you that when you have conditions that with the wild type plant you reach a kappa number of 15, and the kappa number is the amount of lignin in the pulp after the pulping, that the transgenic lines will have a kappa number of 13, and at the same time they will have 1 to 2% point more crude pulp. So this is certainly interesting for the pulp and paper industry. So I've shown that gene discovery and very fundamental research in Arabidopsis can lead to very interesting and promising data for applications in biorefinery and in pulping. The second story I would like to tell is about genetic gene editing with a focus on another gene in the pathway. So cinnamyl-CoA reductase catalyzes an important step in the production of these monolignols. Using CRISPR-Cas, we can knock out the complete gene. We can make it dysfunctional. CRISPR-Cas is a system that you introduce in the cell. The system goes to the target gene, in this case CCR. It cuts the DNA split. The biological system will repair it, and sometimes it makes a mistake, and therefore the gene will be knocked out, will not be functional anymore. And so if you knock out the gene, your plants will be very small. They will saccharify very well, but they are too small, not interesting for applications. But it does not mean that CCR is not a good target for applications. And I will explain you why in the next slide. So our tree that we use is a hybrid tree. It's a combination between Populus alba and Tremula. Actually, all trees are hybrids, like humans. And so that means that we have one copy of the CCR gene from Tremula and one from alba. And the gene editing machinery has made, and the arrow apparently points wrongly, but a single base pair insertion in one copy, which makes this copy defective, and it makes an 8 nucleotide deletion in the other copy. And this makes also the gene defective, and that's why we have these very small plants. But we had also plants which have one insertion in one allele, making that allele defective, and a -3 nucleotide deletion in the other allele. And 3 nucleotides is the same as a codon. So that means that if you translate in silico this new edited allele into the protein sequence, you will create a CCR protein, but with 1 amino acid less, and also with 1 other amino acid, because the 3 nucleotide deletion was not completely in frame with the reading frame. Okay, but you do create a functional CCR protein, and those plants that are gene edited, they are normal in appearance, but they have reduced lignin, because if you peel off the bark, you have this typical red xylem coloration, which is a hallmark for CCR deficiency. The plants have 10% reduced lignin, and they have a significant improvement in saccharification efficiency without or with an acid or an alkaline pretreatment. And so these trees are now in the field in collaboration with Sweet Tree Technologies in Sweden here. A third example is how we can make completely novel lignin polymers that are not yet potentially found in nature. And this story also starts from our co-expression analysis. So we identified a new gene that appears to make a protein called COSY that takes 6-hydroxyphenylwyl-CoA into scopoletin being a coumarin. And 6-hydroxyphenylwyl-CoA is derived from phenylwyl-CoA. It's a part of monolignol biosynthetic pathway with an enzyme called F6'H. So how did we discover the function of that gene? Again, we used comparative metabolite profiling. So we analyzed the metabolites that were increased in abundance and decreased. So in red, those that are increased. And using mass spectrometry, we try to unravel as many structures of compounds that accumulated and disappeared. And by comparing the structures of the red and blue boxes there, we hypothesized that 6-hydroxyphenylbyl-CoA was converted to scopolamine, and this we could then actually show in an enzyme assay. Now, the neat thing for this talk is that scopolamine shares structural similarity with a coniferyl alcohol, so we potentially can use it to incorporate it into the lignin polymer. I'm going to give some more details here. So this is again the 2 steps from phenylbyl-CoA, a pathway intermediate in monolignol biosynthesis, towards scopolamine. And this is coniferyl alcohol. So where does it share structural similarity? Well, scopolamine and coniferyl alcohol, they have this para-hydroxy function. This is needed to create radicals, biolaccases and peroxidases. The molecules have the 7-8 double bond, which generates the ether linkage in lignin, and ether linkages are the most frequent units in lignin. And scopolamine has this additional advantage that it has an extended conjugated system. So that means that if we can boost the abundance of that molecule in lignifying tissues, if it incorporates into the lignin, it will create ether linkages that are more susceptible to alkaline degradation. So our objective was to produce scopolamine, which is normally only present in roots. Objective is to make it in stems. And that's why we expressed F6'H and COSY, these 2 genes, under the control of a strong lignify, a promoter that lignify, but is expressed in lignifying cells. And analytical tools then showed that the plants really they produce more scopolamine, and also its glycoside scopolamine. And you can even see the incorporation of scopolamine in lignifying cells in Arabidopsis based on its fluorescence characteristics. So these plants, they do incorporate scopolamine in the lignin polymer, as demonstrated by NMR collaboration with John Ralph's lab. At the right panels, these are NMR spectra from the transgenic lines, and at the left of the wild type. And at the left bottom here, you see a difference spectrum. It subtracts those 2 spectra, and therefore you only see the differences. So the red contours reflect the scopolamine molecule incorporated or attached to lignin, because they are the same contours as NMR spectrum from pure scopolamine. Now, finally, upon an alkaline pretreatment of this biomass of Arabidopsis, you can see we got a 40% increase in saccharification efficiency. Again, this was very interesting because the plants did not have yield penalty. Therefore, again, we translated that research into poplar. We have trees now growing in the field that produce scopolamine, and so far they are growing well. And if you want to know more about these trees, we have a poster from Nette de Ridder hanging on the hall here. And we have also another poster by Chantal Anders, who is following up another gene derived from this co-expression analysis. So, in conclusion, we can make trees that are better for industry and for the environment. So, instead of making trees that have a higher biomass yield, we make trees that have a higher yield of the product you can make from wood. So we can save chemicals, we can save energy, so we can make more product from a plantation. And that should, I hope, reduce pressure on harvesting native forests and the associated biodiversity. And in this context, I think it's important also to say that genetic engineering and gene editing, they are not alternatives to classical breeding. Genetic engineering and gene editing are tools that create extra genetic diversity and phenotypic diversity. And these tools need to be integrated with conventional breeding to make trees that are suited for the pulp and the biorefineries. And I would like to close, of course, by saying that I did not do this work alone. The publications that I discussed are listed here. Also, the groups that have been very important in that research, even if I did not show all the data that these produced. This is my group in Belgium, so you will recognize Nette and Chantal, who will be presenting a poster here, and Tatjana Mota, who has been much involved, or well, responsible for the work on the poplar field trials that is not published yet. Thank you very much for your attention.
Thank you very, very much indeed for that. Would you mind answering a few questions, if there are any questions from the audience? I'll start by saying one of the remarkable things that your work and others has shown is the flexibility that trees show for changes in lignin. One might have thought that small changes in lignin structure would be very detrimental to the growth of the trees, because the main units in lignin are quite conserved across many trees. Do you see any opportunities for changing the lignin to improve the properties of timber in trees? You've talked about refining from them, but are there changes that perhaps we could make to make the timber properties better?
It's a very difficult question. So let me come back to the fact that we can modify lignin composition. Actually, we do not really know in advance whether the modifications that we bring in the lignin polymer are going to be accepted by the plant or not. And it depends, of course, on the amount of a new unit you incorporate into the lignin polymer, but it also and on the level of gene downregulation, as I've shown with CCR, but also the position in the pathway where you make the modification. If we make modifications in the gene CCR, we always have a higher chance of having a yield penalty, because apparently if you block that particular step, some metabolites accumulate in the cells, and some of these may be toxic. Now the plant cell has a system for detoxification. It will put the glucose on the molecule and store it in a vacuole. But other steps like CSE do not appear to provoke such a toxic path accumulations. So depending on where you block it, you have a different response by the plant. Yeah, but that wasn't an answer to your question. and the reason this, but it's actually a very important point, is that many researchers, they stay more or less in their research field. So the chemists stay in their chemistry field. They have a lot of knowledge. The biotechnologists, they know about modifying genes and gene expression levels, etc. But we need to bring these people together, because many chemists might have brilliant ideas, but they may not realize whether we can, or they start from the existing lignin, so they think, okay, we'll decompose the existing lignin fraction, I can maybe make products from that, but I may not realize what is the potential with lignin engineering. So we need to kind of come closer together and start discussing. I think that's very important.
Yes, yes. Was there a question from the audience? Yes, please. We have a microphone coming for you. Yes, just coming.
Göran Gellerstedt, KTH. I have a question concerning lignification. When you talk about all these various monomers that can be incorporated into a lignin, I guess that the solubility of these monomers must be quite different in an aqueous solution. Can you comment on that, how this nevertheless gives you lignin with these types of incorporation monomers?
Well, in the cytoplasm, the monomers are soluble. And so it is true that any molecule that can be oxidized to a radical potentially can couple with another radical in the lignin polymer. But the first requisite is that this molecule needs to be able to travel through the plasma membrane. And people in the past thought that there were specific transporters to do that, proteins in the plasma membrane that transport these molecules. But Greg Beckham and others have shown that some of these molecules, when they have the right properties, they can just diffuse through the lipid bilayer into the cell wall. So even for example, if we would have a molecule that we would like to incorporate in lignin, for example, ferulic acid, this will not travel through the plasma membrane. So there will only be minute amounts of ferulic acid traveling to the wall. And the same is with glycosides. So from the moment we produce also scopolitin, so we overexpress the molecules, so the genes that made scopolitin in the cytoplasm. Part of that scopolitin has the property to transfer over the plasma membrane into the cell wall, but part will be glycosylated because the plant doesn't like such a high levels of scopolitin, and will glycosylate it to a molecule that will be stored in the vacuole. So there are, if we are thinking about what molecules we would like to incorporate in the lignin polymer, we first have to think, will these molecules be transported over the plasma membrane without the use of transporters, just free diffusion? If this will not happen, we will not be able to incorporate them in the lignin polymer. But is this an answer to your question on solubility? Yeah, okay.
Thank you.
Okay, I think we should move on. Thank you very much again for your lecture. Yes, and you made another good point at the end there about collaboration being really important in these developments. And we've seen from Wout's work how molecular genetics and manipulation of plants allows us to see the potential for changing lignin structure. But the full potential of the application only comes if we understand the lignin structure in great detail. And I'm going to ask Professor John Ralph to give his laureate lecture.
So thank you to Marcus Wallenberg and your incredible foundation. Thank you for all being here. So I'm going to dig into a few of the details of some of the work that Claire and Wout just talked about. Long title, sorry. But first I would like to highlight how important the collaboration has been with this quite ridiculous slide of the productivity from the two groups. It really has interacted in an unbelievable way to advance things that neither of our groups could have done by ourselves, and I hope you caught all of those. Also, very happy that on the way, we've gotten to work on all kinds of projects that I'm not going to talk to you about, with wonderful collaborators all over the world, even occasionally getting cover articles that are always nice, and I wish I could talk to you about all those things. The thing I do want to highlight is, first of all, the technique that came out of the Ghent lab. They cleverly called it lignomics. Some of you will understand the connection there, but it's to gain an insight into early lignification, what's happening early in the process, either during early plant growth or early in the lignification process itself. And a whole group of people in both groups have been involved in this work for a long time. The details. So this is a little piece of a very old chromatogram showing some of the components that come out of that. You can start annotating them from the mass spec. I'll zoom in on one of them to show you that this product, for example, is an oligolignol that started with coniferyl alcohol, started with synapyl alcohol, coupling to it in this beta-5 way that Claire mentioned, and then another coniferyl alcohol comes along, adds to the free phenolic end of that unit, and makes the beta-O4 linkage. So this is a trimeric unit on its way to the polymer that grows from the phenolic end. The key for these things is you have to prove at least one or two examples of those, otherwise you're just speculating. So we prove these by synthesis and by NMR, and I'll talk about that in a bit more later. Just an application early on for a gene that wasn't mentioned yet. There's a O-methyltransferase on the way to making synapyl alcohol from the coniferyl alcohol pathway. And what happens when you crank this one down? You can either back up the whole pathway to coniferyl alcohol, or perhaps you can make the intermediate product and make a new monolignol that we hadn't seen in the plant material before. And so the question becomes, does this 5-hydroxyconiferyl alcohol, is that a lignin monomer? And does it produce these 5H units in the lignin, in which case that would be novel? So the polymer again, the profile again. The COMT down-regulated material has peaks go up and down, but it also has some new peaks, and I'll home in on one again. And you can start annotating these peaks, done in Wout Boerjan's lab again. One of these, for example, is where coniferyl alcohol has added to 5-hydroxyconiferyl alcohol. Again, because that's a new product, you really must prove that. So you do that by radical coupling reactions or by synthesis and NMR proof afterwards. Oh, sorry. And then the next step after that is to understand whether it gets incorporated into the lignin or not. And I won't go into this at the moment. We'll talk about NMR in a little bit. But basically, the NMR spectra of the side chain of lignin contains these beta ether units and beta 5 and everything else. Does not contain those circled red units. These are unique when you knock down that COMT gene, knock down that part of the pathway, and it's because of the incorporation of those units, those novel units, that new monomer into the lignin. For the young researchers here, I want to point out, sometimes, yes, that is a little peak. Identifying tiny little peaks is also important to some extent. And in fact, you can get a whole publication out of just identifying this tiny little peak. Now, before you get too excited, there's some things that come with that. First of all, it's only important because it's actually quite interesting in revealing details about lignification itself, and it's only important once it's really authenticated. This is a very strange structure, right? So you really do have to authenticate those components. And I do want to make the plug for organic synthesis again, which might be disappearing a little bit as a discipline and is still as required as ever. It's because, in this case, why this is novel, is normal monolignals do beta-beta coupling and make these kind of products. This thing looks like half of that, but something else has happened. And it implicates the idea that you've made a synapyl parahydroxybenzoate conjugate first, and you've used that conjugate for lignification instead of the monomer, once it's authenticated. So the implication, in other words, is that you've taken your monolignol, you've got parahydroxybenzoic acid, activated form actually as a CoA, you've made this intermediate conjugate, and you incorporate that into lignification then by the normal process, and you make a lignin polymer with these pieces hanging off the edge. Interesting by itself, but that means there's a gene or an enzyme involved that we didn't know was part of the pathway before, and maybe it's useful. Why might it be useful? Because it might be useful, first of all, to understand the process, but also to manipulate the levels of a component like this. And why might we want to do that? We got quite a bit of press for a very simple project where we just took these parahydroxybenzoates on poplar and started making a pharmaceutical from it, paracetamol here, or acetaminophen, Tylenol in the US. And like I say, this attracted public attention for something that was a very simple project for us. But let me show you why we think that's important for the kinds of things we're thinking about for replacing replacing fossil fuel materials. Currently, Tylenol, paracetamol, is made from coal tar, crude oil that needs a lot of refinement to make benzene. You spend then 3 very good but massively industrially complex steps to just put the phenol on that aromatic to make phenol. Then you nitrate it, you throw away the piece that wasn't useful, you reduce it, you acetylate it, and you make Tylenol. But look what nature has already given us. It gave us this parahydroxybenzoate sitting there on poplar lignin. And if you look, nature's functionalized this beautifully. It already has this phenol on it. We don't have to go through those silly steps up the top there industrially. This maybe looks a little bit weird, but there's some beautiful chemistry that affects that. Well, so first of all, the parahydroxybenzoate in the poplar lignin, you can just toss it in ammonia and get a very good yield of this amide. Vitali in the lab got this yield up to 98%, so very viable reaction. And then a simple bleach reaction turns this thing around, makes paraminophenol, which is actually a commodity chemical of high value and high volume in its own right, and then you acetylate it the same way. So I think you can see that process is so much simpler. You wouldn't think about going back to the fossil-derived pathway if you knew this existed in the first place. How short-sighted are we to neglect what nature has beautifully functionalized for us already? And I think we're starting to appreciate that. And there's some groups all over the world. Catalin Bata, for example, is making all kinds of products from lignin now that I don't think were considered previously. I want to talk about the NMR component. President Obama gave us this nice machine. Grateful to him during the bioenergy renaissance, if you like, in the United States. I wonder if you're aware that you don't need to isolate cell wall fractions anymore. NMR is usually considered to be useful for pure proteins or small molecules. We can take whole wood now. We have to grind it up quite finely, but we swell it in a solvent and can run solution state NMR, quite high resolution on that. And I'll show you why that's sort of useful. Oh, first of all, yes. If you take this kind of sample to an NMR department and you turn the tube upside down and it doesn't flow, they will tell you what you can do with your sample. It won't be nice. But it's extremely useful. So these are, in some ways, horrible spectra and yet unbelievably useful profiles of the cell wall component. In the aromatic region, just the aromatics show up. So pine or softwood lignins have just guaiacil lignins. They have the 3 peaks. I won't go into explaining why that is. But basically, that's the little profile for a guaiacil lignin. You take something like Arabidopsis that Wout mentioned. Suddenly it has syringal groups in it, guaiacil groups, and these H units Claire mentioned also. You take poplar, S and G, also has the parahydroxybenzoate in it that we're making that Tylenol from. And then grasses have a lot of paracumarate on the lignin, as well as ferulate on the cell wall. It's mostly on polysaccharides, but all of those things get profiled. Make one point that if you think you have some component in your cell wall prep by, for example, seeing a peak here, the nice thing about NMR is you need all of the data from that component. So in other words, if you suspect that this peak might be paracumarate, you better see those other pink peaks too, or it's not paracumarate. So it's very diagnostic in that way. In the side chain region, same spectra. Overwhelmed by polysaccharides, but you can see the way these monomers connect. So beta ethers and phenylcumarins and so on. I won't go into this, but you can see all of those components in these complex spectra without, again, having to isolate the components. And you can actually quantify your polysaccharide distribution as well, and I won't go into that. It got better. It was never quite quantitative. Last year at the MWP, Stefania gave a nice talk, a young researcher's talk, on an improvement that allows the quantification to be done much more rigorously. And I won't go into that either, but just know that it exists. All right, to illustrate NMR just a little bit further, I'd like to do this in a slightly educational way. Forgive me, the young researchers here, I hope this is useful. Talking about a new gene that we've discovered on the pathway that's eluded us for a long time. We've already talked about monolignol conjugates, the parahydroxybenzoate and poplar, this paracumarates on grasses. Ferulates I'll talk about later, but the acetates that are on all hardwoods and all grasses eluded us as a gene for a long time, till Nondagula came along and somehow targeted this gene and has made successful transgenic poplars. And I'll use this as a case for illustrating the power of 2D NMR, I wish 3D NMR, but also the model compounds again for understanding what is happening in that plant and documenting those changes. So here's these horrible Rorschach tests again of wild-type poplar. These are the beta ether units and so on, all colorized very nicely from normal lignin. The transgenic with the acetates created, if you are good at doing these YouTube spot the difference experiments, you'll see immediately some differences, but I want to show you where those come from. So first of all, you can assign them. How do we make those assignments? We make them by, well, 3D NMR, which I can't show you, but mainly by model experiments. So you take, for example, this beta-5 coupled coniferal alcohol unit, a model for that. You run the data, run the NMR, very simple, very sharp. You zoom it back onto the polymer, and you see that we've explained the alpha peak of that B unit, the gamma peak there, and the beta peak. Those things match exactly when you have good models for the polymer data. Then what happens when you acetylate this material, or make an acetylated model, you run that back, and you realize, aha, I've just explained these new, the B alpha at the bottom is in the same place, but we've just explained a couple of new peaks, that one way at the top and the one in the middle. In other words, when you acetylate a lignin unit like this, the B beta peak moves up there, The gamma peak moves down here, and the alpha peak doesn't move. And there's also some of these N groups in lignin, and those things move and give that new peak as well. So very quickly with bottle compounds, you realize, okay, we've explained a couple of the new peaks in the spectrum of the transgenic. If you make beta ether models, so I won't show you that, but you get to explain another set of these peaks and that N group. So basically with those three models, we've explained basically all of the new peaks that you see in the spectrum and assign those to the fact that acetylation of that lignin naturally has occurred. Okay, switching gears a little bit. Wanted to tell you a little story about something we tried to do to teach nature something new, to make lignin that was a little better for our industries. And it's one of, well, I'll explain it in a second. So poplar lignin model, we've seen it before. Its beta ether bonds are the weakest bonds, but they still require really harsh conditions to cleave those up. And you're trying to cleave the polymer into smaller units to get it out of the way, to dissolve it in the pulping liquor so you can get at the cellulose. Wouldn't it be great if we made more easily cleavable bonds in the backbone of that polymer? How might one do that? Here's one way. The pathway that Claire showed, coniferol and synapyl alcohol, in that pathway is ferrulyl-CoA. If you had an enzyme that would connect those two, you could make this monolignol conjugate, a bit like those pyrohydroxybenzoates. And so what? Well, we know a lot about how ferrylate couples. It's very like the monolignols in the way it does its radical coupling reactions. So it would incorporate at both ends. You incorporate it, sorry, I'm artistically challenged. This is my way of showing it incorporates into the polymer. But what you realize you've done is you've incorporated these ester bonds into the backbone of the polymer. So you've made a polymer that's sort of modular. It's designed to fall apart into pieces very easily. Yeah? That was the idea. Basically, you make some lignin with these components integrated and it falls apart under much milder conditions very easily because of the ester bonds. This got into Science, that's nice, not on the cover. And in it, we did note that these are plants that are designed for deconstruction. You have to test that, of course. Again, Troy Runge's group, Shengfei, did beautiful pulping studies, the most boring graph you've ever seen. You pulp to a given residual lignin level, depending on the kind of pulp you want. And at every level, the transgenic gives a 2% higher, 2 percentage points higher yield of pulp. It's very substantial for a pulp mill and worth a lot to that mill and to the industry, in fact, that kind of improvement. This has been licensed and plants are growing in Brazil. Just by the way, we wrote, these plants are designed for deconstruction. You can't always write your own headlines though. The news headline was cheaper fuel from self-destructing trees. What? So the internet lit up for a little bit. Please be assured these plants don't care. These are not materials that fly apart by themselves. They fall apart a bit easier in caustic soda, which is not very common in the natural environment. Just by the way, sorry, we've got better zip genes now. I'll just point out from NMR profiling again, we've got some materials that have much more ferrylate in. So we've got new genes. These materials have not been tested yet, but we're looking forward to those because they should be way better. But I said we were arrogantly trying to teach nature a new trick. Millions, hundreds of millions of years ago, she was already doing this. We didn't know it. This is good for the young researchers too. So first of all grasses already contain this kind of lignin, zip lignin. Half of the hardwoods, most of the ones that we choose to pulp, as it turns out, contain this kind of lignin. How the heck did we not know that it already existed? Two things, we weren't smart enough. We actually had the techniques. We potentially could have discovered those with the metabolomics and the NMR. Simply no one had thought to ask. And there was no analytics to determined that those monolignal conjugates were made, but that they were used for lignification too. And I won't describe this because it's too complex, but Fashuang Liu came up with a lovely method to cleave ethers in lignin, but leave esters alone. And so this is a method that makes these monolignals back out of lignin again, an analytical method, but it leaves these conjugates intact as well, from which you can prove not only that you had this conjugate involved in lignification, but that went into lignin itself. Beautiful methodology that didn't exist until this zip lignin project. All right, I want to thank so many people, none of whom you can do justice to, across the world. It's been a marvelous ride. It's only just starting for you young people. You have big challenges on your plate for the future of this planet, and good luck. Whoops. Thanks.
Beautiful demonstration of analytical techniques together with biosynthetic experience. Thank you very, very much. I think, yes, we are. No, you don't want questions, right? Okay. Yes, out of time. Okay, so now we're going to have the announcement of winners amongst the young researchers who are here at the symposium. So I'm going to hand over to Hani Calderas to tell us about this.
Okay, so I'm coordinating the Young Researchers event, and that's a program, a scientific program for young researchers that already started on Sunday. So the young researchers have gathered around 30 young researchers from all over the world. We are delighted that we have a representation from not only Europe this year, but also from Northern America and South America. And they represent 8 different countries and 18 research institutes or universities. And the scientific program started already on Sunday with some scientific discussions with the laureates. That was highly appreciated from both parties, I think. And then continued yesterday with external research discussions and the 3-minute pitch presentations. And associated with that, we also had a competition, and I'm here today to announce the winners. And as also mentioned a couple of times, we have a poster session. So once you have the break in a couple of minutes, you are welcome to join the poster session. Please visit the researchers and ask them questions, because they put a lot of effort in compiling these posters. And the presentations yesterday, they were all recorded and will be uploaded to the YouTube channel, the MWP YouTube channel. And we will post a link both on the webpage and on the channel. So please take a look at those too when they are published. And we will actually show, after I've announced all the 3 winners, we will show the winning pitch presentation. So Joris, if you could please join me on stage. We'll start with number 3. So the 3rd prize for the pitch presentation goes to my former research group at KTH, Åsa Järlhagen. Okay, 2nd prize goes to Julia Assi from the University of British Columbia in Canada. And 1st prize goes to Tinkara Bisjak from SLU in Sweden. Yeah, so we will shortly have the break, but please remain seated and view the pitch presentation.
Hey, I'm Tinkara, and I'm a PhD student at the Swedish University of Agricultural Sciences, and more specifically at Umeå Plant Science Center. As I'm sure you all know, wood biomass is extremely important as it can be used for so many different purposes. If you try to imagine a relaxed evening at home, it could, for example, include eating dinner with your family around the kitchen table, or maybe reading a thrilling book before you go to bed. All of these activities include the use of wooden products, from furniture, books, to in some of our cases, even our houses, which are mostly made out of wood. However, the forest land is limited, and one idea to try to get more wood biomass per the same amount of forest is to increase the plant growth. One method that has been used is to fertilize the forest with inorganic nitrogen, because the main nutrient that is limiting the forest growth of the boreal forest surrounding us is nitrogen. This is an effective method. However, it can lead to certain negative environmental effects, such as greenhouse gas emission or soil acidification. So a big idea out there, and the question is if this could be maybe supplemented or even replaced by using nitrogen-fixing bacteria that are already present in the environment. But very little is known about this bacteria in connection to boreal forest conifer trees, such as Scots pine. And that is what my research is about. So nitrogen-fixing bacteria are very diverse. They're present in 10 different bacterial phyla. However, they all share this intriguing ability to fix nitrogen. The enzyme responsible for that, you can see it here, is nitrogenase. And it's capable of converting the nitrogen, which is present in the atmosphere, into ammonia, which can later be assimilated into the plant. And my research shows that these bacteria are actually also sitting inside the needles of the conifer trees, such as Scots pine. So next month, when you bring home a Christmas tree, you might think about the fact that you've also brought with it a lot of nitrogen-fixing bacteria. I also wondered if these bacteria are actively fixing nitrogen while they're sitting inside of the needles. And the results show that yes, they are actively fixing nitrogen while they're just sitting there. And even if the forest is fertilized with inorganic nitrogen, their activity is still the same. In addition to nitrogen fixation, these bacteria possess certain other plant growth promoting properties that can additionally help the plants grow better, as they can influence the plant nutrients, hormone levels, and even pathogen protection. So why not research more about these nitrogen fixing bacteria and see if they can be used in the future to sustainably promote the growth of forest trees? Thank you.
Okay, so that concludes the first part of our symposium. Now, one of the goals of the Marcus Wallenberg Symposium is networking. We have, as we mentioned, a lot of young researchers with posters here. We have a lot of people doing research in universities and a lot of industrialists. So now we're going to have a break for an hour, and you're encouraged to meet some new people, make some new connections, and who knows what will grow out of those new connections. So we'll return here in 1 hour to continue the presentations.
Dear laureates, dear guests, welcome back. I would also like to welcome back the audience we have online. I hope you all have taken the opportunity to see the posters, talk to the young professionals during the break, and networking and build contacts. My name is Monica Ek, and I'm a member of the Marcus Wallenberg Prize Selection Committee, and I will guide you through this second part of the today Marcus Wallenberg Prize Symposium. So this last part will be divided into two parts, and the first one will be about lignin innovations. To recap the morning, we have listened to Claire, and she was setting the scene for our laureates. And then we have these amazing presentations of this groundbreaking research of our laureates, Professor Boerjan and Professor Jon Ralph. Lignin is a very interesting, amazing area, and it really deserves this prize this year. So, but to start this second session, we will listen to innovations in the first part, and our first speaker is Monica Österberg, who will talk about From Craft to Craft. So welcome, Monica.
Thank you, Monica, and thank you, organizing committee, for inviting me here today. It's really a great honor, and I'm so glad to be in this session and talking in honor of this of our laureates, Wout, Boerjan, and Jon Ralph. They are really iconic scientists in the lignin community. So this is really a great honor for me today. So I will talk a bit about how to extract value from lignin. So we have heard now that you can do gene modification of the tree to get a nicer lignin, or you can modify the biorefinery. But I'm a very pragmatic person, so still we have to live with that the 90% of the lignin available, technical lignin available today, is craft lignin. So how can we make added value out of this? We heard in the morning that the craft process, for example, is it's optimized for getting nice cellulosic pulp fibers. So the lignin is suffering a bit. So it's a bit difficult to get value out of this. So there's different ways. The classical ones, fractionation or chemical modification. These are all many-step processes, so that usually adds cost to the process. Also nowadays, if you do the chemical modification of lignin, you might affect its biodegradability, and also in EU you affect its classification, so it goes from a natural material to be classified as a plastic, which is a bit of a challenge. There's still a lot, I don't say it's bad to do chemical modification, there's still a lot of possibilities. But we wanted to make it simple, just get these nice spherical particles that are water dispersible, and in this way make lignin a bit more easy to use. Of course, we will afterwards, in the later stage, hear a lot also that we can modify the biorefiner process, and maybe this is the future, but until then we have to make some value out of the craft that we have. So what we have done is that we have developed a scalable, economically valuable method to produce these lignin nanoparticles. It's very simple. You take the lignin, you dissolve it in an organic solvent, and then you pour it into water, and spontaneously it forms these spherical particles. And we can use different kind of lignins, and it's also, there's now a startup company, Lignosphere, who is trying to make money out of this, so trying to commercialize the application possibilities of this one. We have done these particles, studied how to make them, how to affect the particle size, and then how to apply them in different applications, ranging from microcapsules for drug delivery or hybrid particles for energy storage, to adhesives or functional coatings or even foams. I will not have time to show all of this. I will give a few examples. And what I will start with is based on the lignin particles' ability to stabilize emulsions. So you know, if you want to mix an oil phase with a water, you need some, usually you can use a surfactant, a small molecule surfactant. If you instead of this small molecule use a solid particle, it's called Pickering emulsion. And this lignin nanoparticles are very good to stabilize this kind of emulsions. And you can use the emulsion as such. For example, for cosmetics, it's quite nice that you get also UV protection in this. So for sunscreens, there's some research ongoing with that. But we used it as a template to make materials. So we have done like waterproof biodegradable films or foams, or use this for drug carriers. I will talk about only this example today. So how do we do this? Use this Pickering emulsion template to make the composites. We have a few cellulose chemists here that have worked a lot with nanocellulose. So you know that if you nanocellulose makes very nice nanopapers, nanofilms, but they are very sensitive to moisture. So there's a lot of work how to make them more hydrophobic. If you just add something high, so what we wanted to make these nanopapers more flexible and also more water resistant. So we wanted to combine with another biodegradable polymer, polycaprolactone. But these two, polycaprolactone doesn't dissolve in water, so we need a compatibilizer, and we used lignin nanoparticles as this. So we made a very stable emulsion, and if we hot press this emulsion, we can get this nanopaper or composite film. And indeed, if we look at nanopaper, it's very, very strong, but it's also very brittle. And if we do this composite together with the lignin nanoparticles, we can increase the flexibility of the film a bit, and it has quite good dry tensile strength. Usually, you can either get good dry tensile dry strength or good wet strength, but not combining this. But with this, we actually got very nice increase in the wet strength. So we soaked them in 24 hours in water and then tested again the strength. And if you see here, cellulose is very poor, but then with this composite, we get a quite nice strength. So this composite film, it retains its strength even when it's wet. But I was talking, I want to have this recyclable and biodegradable material. So what happens is, like, our hypothesis was that since we are only mixing, we are not making new covalent bonds, will this be still biodegradable? And indeed, we can retain the biodegradability of this material. And the reason is that we are not adding new covalent bonds. Of course, it's not going to 100, and the reason for that is the lignin nanoparticles. Anyone who has been trying to biodegrade lignin, it's one of its roles in nature is to slow down this degradation. So it is degradable in soil, but in water it's not. It's very slow. But I personally don't think that it's a big problem. So it's still good. Then I will go to a few examples of how these lignin nanoparticles can be used to make functional, multifunctional coatings. The first one is to make this fire and water resistant coatings for wood. So this stool there that you can see, it has been painted with this system, with this lignin nanoparticles. So what we did is we combined the lignin nanoparticles with an epoxy, and then here we are forming new covalent bonds. They are reacting, and then we get a very quite abrasion resistant coating that is then also resistant to sunlight, solvents, and stains. So it's a multi-resistant coating. What is interesting is that it is hydrophobic, so it's protecting from stains and from water, but it's still so thin, the layer, and since it's particulate, it's not. It's also breathable. So if you know that, if you want to keep the good breathability of wood, you don't want to paint over it with a thick lacquer, for example. So you can see, and this is shown just with this moisture buffering value. But epoxies are skin irritants, so we wanted to go for a bit milder approach when we wanted to make functional textiles and also barriers for packaging. And there we wanted to be able to recycle the material, and also that it would be safe chemicals. So then we went into using fatty acids and enzymes for the modification. And I will show you two examples. One, where we want to, for the textile, you want to keep the breathability, so you want to have a thin layer. For the packaging, you do not want to have the breathability, so you want to have a barrier against the gases. So first, this thin multifunctional textile coatings we used. Here we chemically modified the lignin with lauric acid or tall oil fatty acid, and they formed still nice particles after this modification. But we were able to do this functional textiles with layer by layer approach. We got some typical UV resistance and this antibacterial that you usually get when you are using both lignin or tall oil fatty acids. But it's also kind of super or almost super hydrophobic and anti-staining. As you can see, like if you drop wine, coffee, or milk, it will stay as droplets and easily roll off. And this was breathable. We compared both air permeability and water vapor transfer ratio for like these different modified lignin nanoparticles, and it keeps the same level as the reference unmodified cotton. But when we made a bit thicker layers for this packaging, we can again make get nice barrier properties. So just here is this system. So we see that we have quite low oxygen transfer ratio and water vapor transfer ratio. And then here are like examples what is needed for different food packaging. So as a final example, like can we overcome the 50 shades of brown that we usually have when with lignin? And yes, indeed, we actually can that. So by making acetylated lignin nanoparticles, we can form very, very small particles. And then if we spread just a monolayer of these particles on glass, then water, it becomes super hydrophilic. So it will actually form this kind of a homogeneous water layer. And that means that you can see through it. While normally if you have a contact angle of 20 to 30 degrees, you will get these water droplets and they scatter light and you cannot see through it. So we can make invisible anti-fogging coatings. And if we make thicker layers, we can do this structural coatings. So we can get from this boring brown to bright purple or blue with different thicknesses. So to conclude, I think lignin has a bright future. We will hear examples after my talk about it. And I talked about how to control the morphology of lignin. I think we can do a lot with that. But obviously, it's important to understand the chemistry, be able to measure the chemistry, and also like controlling it, as our laureates have shown during their research. And of course, then I think we will hear also about scale up, and I think sustainability is also important. So with that, once more, thank you for listening.
Thank you, Monica, for these very interesting examples of what you can do, and also with the lignin nanoparticles. Do we have a short question for Monica? Anyone?
I hope you hear me. Thank you very much for this very inspiring talk. Tina Piatnik from Lansing AG. I was wondering, the functionalized textiles that you showed thank you so much, with the coatings on it, did you do washing tries on them? Did the coatings maintain when you do washing?
Surprisingly enough, yes. I don't, because our idea was, or our hypothesis was that it would be washed away, and our next way was to do enzymatic linkaging, but for some reason, actually, it was stable also for some washing. Maybe not hundreds of times, but.
Okay, I think we have to continue in the program, but all speakers will be here for lunch also after this session, so we can continue asking questions in the end. Thank you, Monica. So, our next speaker is Hanna Karlsson, and Hanna comes from Valmet, and she will talk about state-of-the-art in industrial lignin separation. Welcome, Hanna.
Thank you very much, Monica. It's an honor to be here today, together with all these excellent people. So, my name is Hanna Karlsson, working as Business Development Manager for Lignin Technology at Valmet. I've been working with lignin since 2008, when we acquired the LignoBoost technology. I began my career as a process engineer, and during that time I had the great opportunity to be responsible for the process design, and also part of the startup crew that started up the world's largest kraft lignin production plant so far. I'm here today to talk about lignin production in industrial scale. So I'd like to start by highlighting the key takeaways that I hope that you remember from my presentation today. And that is that the technologies to produce lignin in industrial scale is already available. The technologies to produce valuable products out of the lignin, it's also available. And you will hear more about those excellent products that you can produce later today from the other speakers, and you have heard some of it already by Monica. So if those technologies are available, what's holding us back? Why isn't the markets going to kick off? I'm not going to answer that question, but I'm going to talk about what we have done at Valmet in order to help the market a bit. What you see in this jar here is LignoBoost lignin, And LigniBoost is Valmet's technology for producing lignin from one of the streams in a pulp mill. And for those of you who are not familiar with Valmet, we are technology and equipment supplier for the pulp, paper, and energy industry. We are about 19,000 employees. We build complete plants or factories from the ground and up. We take them in operation and we hand it over to our customers, so we don't have any plants ourselves. So this picture is showing a pulp mill a bit north up in Sweden, and it's a good example of what we can build. And utilizing the pulp mill and generating both more and varied products out of the same amount of raw material is one of the important things that we do, that we all do here, I would say. And lignin is one of those products and has also been a focus area for Valmet for many years. Before I continue with the real topic of today, I just want to highlight kraft lignin a bit. Kraft lignin is the type of lignin that you get out from the majority of all pulp mills today. And based on the amount of kraft lignin that is available, there's also a lot of lignin available. And it's quite easily available. It's already there, separated from the tree in a pulp mill. Another positive thing with kraft lignin is that the wood has already been harvested. So you don't need to cut down more trees in order to get the lignin out. And it doesn't compete with agricultural resources in that way. You could debate if pulp does, but the lignin from the pulp actually doesn't. But enough about that. I think you already know this. So, what is state of the art in lignin production? I cannot give you an answer that will cover everyone's view, but I can explain why it means to me and what it means to Valmet. Basically two things. Plant integration and infrastructure is the first thing. And tailoring lignin for the right quality is the second important thing. And I'll try to explain what I mean with that. So plant integration, or rather mill integration, since I'm talking about a pulp mill here, is about integrating the lignin production plant in a sustainable way, both environmentally and economically. So meaning closing the loop, minimizing water, energy and chemical usage as much as possible. But it's also about the infrastructure, to have the infrastructure for the lignin handling in place. And that means transportation and storage at the mill. The physical properties of the lignin could also be of importance, both for the handling at the mill, but also for the end producer, how they are doing in their processes. So things like drying and compacting may also be needed. And then, of course, the lignin needs to be packed in a proper way in order to be exported from the mill. So we are not only talking about the LigniBoost process or separating the lignin from the pulp mill stream. We are basically talking about the lignin mill here. I won't go into details about the process, but if you want to know more, please catch me afterwards. So, making use of the experience that we have gained the past, I would say, 16 years within Valmet is, of course, an important part of the development towards the lignin mill. We have built 2 industrial scale plants, one for the Domtar Plymouth mill in the USA, and one for Stora Enso's Sunila plant in Finland. We have also developed a modular based smaller scale LigniBoost plant, which we called LigniBoost Extra Small. We have delivered 2, one to Klabin in Brazil and one to Mercer in Rosenthal. And this LignoBoost Extra Small has been developed in order to help our customers and in order to help the market. With this plant, our customers could learn about the lignin production, they could produce lignin for application testing, and they could build their own lignin business, or starting to build their own lignin business, without having to take that big investment decisions which is needed, since the barriers are still quite high to do that when the market is like it is. So by that, we think or hope that we are helping the market on its way. So state-of-the-art lignin production is also about tailoring the lignin for the right quality, meaning the right quality for the right application. And whether that application is a high-value product that needs a lot of lignin refining, or if it's a commodity, the quality needs to be the right for the intended purpose, basically. Examples of quality could be improved purity, it could be solubility, or a specific molecular weight, or spherical particles, for example. But what is the right quality? Ultimately, it is the market that decides both the right quality and also the right products to produce out from the lignin. So if the market is the one that decides the quality of the lignin and also which products to produce, how do we actually know what that is, since the market is still very limited? I would say that we within Valmet, we spend as much R&D time at lignin refining and looking into the qualities as we do for lignin production, lignin optimization, and developing the lignin mill. And we have been part of many different development projects together with actors along the entire value chain. Actors like our customers and producers, but also universities and research institutes are a very important part of that development. And in these projects, we have learned about what kind of qualities is needed for a variety of products. We have also got information from our customers and the end users, what they want to have. And based on this information, we have developed several add-on concepts for producing a variety of qualities. And I would like to just mention two of these qualities. One is the ultra-pure quality, which produces lignin with very low ash and metal content. Could be used in a variety of products that needs high lignin purity, basically. And we also have our odor-free concept, which produces lignin with a reduced odor. And the odor, or rather the absence of the odor, is important for many plastic applications, for example. Both these concept or technologies are in a commercialization phase. We are actually right now doing ton-scale tests with the ultra-pure quality. For the odor-free technology, a demonstration plant is being built at RISE facility in Bäckhammar, Ligno City. It's going to be started up during next year. So let's go back to what I mentioned in the beginning. The technologies to produce lignin and to turn it into valuable products, they are already available. So I would say that the barrier is not technical. This picture is an important picture for me. It might not be the most beautiful one, but it shows a belt conveyor transporting the first lignin cake produced in Stora Enso's mill in Sunila, which was a significant milestone in my own career. And for me, this represents that we are doing something real. Thank you very much for listening.
Thank you very much, Hanna, for this interesting presentation of that we already have available lignin if we want to do new applications or new trials. So, is there any question for Hanna? Yes, Göran, please, Gellerstedt. A mic is coming.
Yeah, I was wondering, does it matter if it is pure softwood lignin from the black liquor, I mean, or if it is a mix of softwood and hardwood black liquor that is going into this lignin boost process?
Well, a bit. Every black liquor is different, I would say. Softwood is different from softwood, but also a mixture is different. So it matters. It's just about finding the optimal, process parameters to treat that kind of black liquor.
So that means that every mill has to be optimized if they want to introduce a lignin boost process in order to have a maximum yield of lignin.
Maybe not the mill, but the lignin boost process needs to be optimized for the specific mill. And also, as I talked about, the mill integration, looking at that specific mill, so it will be tailor-made for the mill.
Thank you. We have one more short.
Antti Asikainen from Luke. We have been looking at the market possibilities in a project led by Monica in Finland. And my question is that what would you bet as a first breakthrough application of lignin? There are things like concrete plasticizers and then like surfacing materials. So where is the one who is breaking through, or which is breaking through first?
Good question. I won't bet. We acquired the lignin boost technology in 2008, and we were hoping to have sold many plants. The market is what it is, so I'm not betting.
Okay, thank you very much, Hanna.
Thank you.
Let us... So we are already at the third presentation in this innovation part, and I would like to welcome up Remy Boozer, and he will talk about scaling high-performance material production from wood using aldehyde-assisted fractionation. Welcome.
Thank you, Monica. Thank you, everyone, for being here. It's a pleasure to be at this symposium, especially because this year lignin is in the spotlight, and I have spent a significant amount of my career working on lignin. My name is Remy Boozer. I'm the co-founder and co-CEO of Bloom Biorenewables, and what we do at Bloom is we develop a biorefining technology based on state-of-the-art technology, and I will try to bring you a bit through the journey when we went through, and the best way to start is to show you the spark of when everything started. So this is the exact moment. To be honest, it's staged, as this can be seen maybe, but this is Jeremy Lutebacher, professor at EPFL, Florent Erogel, who is a co-founder and co-CEO at Bloom, and Edna Crestel Santiago, who is our CTO today, also to show that the people that saw this spark are still around this technology, helping it to grow. And what they look at is the difference between if you add an aldehyde or not to pretreatment, and you clearly see the color change indicating that there is something special in here. So a step back, why do we look at all this? It's really the reason that has been mentioned a few times. We need to find other sources of carbon, and wood is an excellent one. And it's actually an underutilized one as well, because cellulose has been in the focus for many years. And as this price also shows, we are moving towards using the other fractions much more, and there is an upside potential there. So what is an aldehyde-assisted fractionation? It's literally you add an aldehyde to an organosolv process, and this will interact with lignin and hemicellulose in ways that allows you not only to get a same quality cellulose that the pulp and paper is producing today, but you get hemicellulose and a lignin that is way uplifted in terms of what you can do. And this is really chemical, so I don't want to show too many structures here, but the aldehyde here in red comes and clips on the lignin and the hemicellulose, making new structures that also, as a company, enabled us to actually patent this very strongly, because these intermediates are now a platform that you can derive in a number of things that have a higher value than what the lignin or the hemicellulose would in its original form. Now, this works on any type of feedstock. We prefer wood for reasons I can dive in later, but ultimately it's a lignin chemistry, so it works on any lignocellulosic biomass, and we have tried many of them, from all the agricultural wastes to nutshells and fancy things. But today we are working on hardwood. So we say that we have basically set the ground for a new biorefining era, and our mission and our passion comes from this middle pillar here, where we really think we need to change something, and we need to change it now. And I think everybody agrees. But then when you go out to the market, which has been mentioned before, the mood is a bit different. Nobody wants to pay for it. And as soon as you don't pay for something, you need to come with other USPs. So we have, as we grew the company, looked at what else is really special, and there you really have the performance, which I'll try to give you a few examples from, and the scalability, because the process is very simple by adding just a component and understanding what the chemistry behind is doing. So we have today a larger portfolio of products, but again, it's cellulose, hemicellulose, and lignin. This is the quality we will look at for pulp and paper type of applications, but also specialty chemicals. We have these protected sugars, which I don't have time to go into today, but you can make solvents and plastics and many interesting things. And on the lignin side, we have the capacity of depolymerizing, so we have monomers, and we have polymers, to make it very simple. And now I would like to dive a bit more into that lignin, because it's the spotlight today, but also give you one or two reasons or proof that we actually outperform existing solutions. So we say we have the only real lignin, and this is really a chemical approach when we say this, is we have the only and first lignin that is extracted and maintains the structure of the lignin in the tree, because we stabilized it with our aldehyde. And this we measure by converting it to monomers. So if you take it from the tree, you get the theoretical yield, and we get very close to this, and this at scale, meaning at over 100 kilos today. We have now brought this lignin to the market in its different forms. We have started actually with fragrances because it was a low-hanging fruit. This has been validating. We today sell these molecules to DSM Firmenich, a large Swiss, now Dutch or half Dutch Swiss company. We have validated coatings, materials, but given the time we have today, I would like just to go into the most recent application that is in the world of resins. And there, I think we really shown that what we do makes sense and can be validated by our customers, because by taking our lignin and just adding water, you can actually mimic the shear strength or the adhesion strength of existing phenol or urea formaldehyde resins, and this is a very unique feature that is actually visible in the chemistry and that has been tried for many years already. So this is a phenol formaldehyde resin that is polymerized, and even if you don't understand or used to see chemical structures, you will see that this is very similar from a structure point of view than what the lignin structures that have been shown today show. And basically, what we do is today, if you extract the lignin, you will actually create condensation within the lignin at the first step of the pretreatment, and you will not be able to create these type of structures within the system. Whereas if you go through the aldehyde-assisted fractionation, you will be able to maintain or to prevent this CC bond to form during the pretreatment, and you can actually form them in the adhesion between the wood, and that's what gives you the power of being at the same adhesion strength. So now, all this does not make sense in the lab. It only makes sense if you scale it. We, as a Swiss company, we have different sites. We close to EPFL for the access to NMR, which again, linked to this price, is essential to do what we do, because we cannot know what we do if we don't have the eyes for it. And as a startup, having an NMR is a bit a shot in the moon. So we work also with partners that allow us to have access to very large equipment, and with this we now have been able to process a ton of wood using our recipe that has been established. And there we are again with a picture of a moment. This one is not staged this time, and this is actually a few weeks ago where we got the first hundreds of kilograms of that lignin. So again, like before, the lignin is real once it gets into your hands, and I'm also very proud to say that most of that lignin has already been sold or shipped and goes to actually validating these applications in many forms and manners. So the story doesn't stop here. So if we look into the future, we have managed to scale this to this 1 cubic meter production facility. And we are today looking at a 1,000 ton plant that we plan to build in Switzerland. And we are very close to have all stakeholders together. One of the elements you need, obviously, for that type of venture is investment, and we have a journey where we were lucky to have the support of Breakthrough Energy Venture in 2020 that helped us really also moving forward and bringing this technology to the scale it is today. And we have now consolidated a consortium around a 12 million Swiss franc round that we will close very soon. There is still an opportunity, so in case you want to participate to the venture, you can come and speak to me. But the kind of deployment of the money will really go to this plant, and we really have the design and looking at the location now to build this. Good. With this, I would like to thank you for your attention, and I hope I have time for a question or two.
Thank you very much, Remy. This is very exciting. I guess we have many questions here in the audience. Yes, we have one here from Jerry.
Hi, thank you. Erik Rönnels from RISE. I was just curious about your aldehydes. You don't disclose it with an R, that's okay, but is it bio-based and sustainable along with the rest of your work?
Yes, so you can make the aldehyde bio-based. We have the chance of having a range of aldehydes we can use, and depending on the one you use, it's easier or more difficult to make bio-based, but the concept is really to go to bio-based aldehydes.
Okay, but you continue to work with a range of different ones?
It's again, we look at the properties you look for are different for different customers. So you need the flexibility. And without going in too much of the details, but you can switch one aldehyde to the other, you can remove the aldehyde, you can really play. Once your lignin is out of the wood and not condensed, you have a certain room to play. And that's where also the different aldehydes come handy.
Thank you.
At the top.
Hi, Sharib this side. What would you comment on the craft lignin that has 90% competition of yours, and how would you see that this technology having processing wood, and currently the price of the wood is increasing? What do you think will be the procurement problems from your startup point of view?
Yes, so I think we need all hands on deck, and so craft lignin will be there. And where the way I see it is today, to make the chemistry, you have to bring in petrobase, so you make blends and formulations to integrate craft lignin into a system. I believe that what we develop will allow to do the last mile, meaning you can do the chemistry that is fossil-based today, even with the same building blocks, and you will have different price ranges for different products. Now, looking at the ultimate goal is to scale. I mean, craft lignin, the same as petroleum-based products, are extremely cheap because they at humongous scales. And our models show there is no reason why this process could not get to these type of price points if you scale it the same way.
Okay, thank you very much. Wish you all the best for the future round, and thank you. So that was our last presentation in the innovation part, and now we are moving to the startup part. And I would like to invite up here Christopher Carrick, and he is from Lignin Industries, and he will talk about lignin, next generation of renewable in plastics, and give some examples how they can be combined with PE, PP, or ABS applications. Welcome, Christopher.
A little bit too excited to go up here, I find. Yes, my name is Christopher Carrick. I'm the founder of the company, Lignin Industries. And I would like to start off with our vision, which is a world where sustainable plastics are the norm, not the exception. I think it bears a lot of mindfulness in that sense. And I think there is a lot of resistance to this change today. What we do in our company is what you see on the picture. We modify lignin chemically, and we produce thermoplastic lignins to be used in ordinary plastics. So this is the most complicated slide I will start with, so bear in mind, this is explaining two industries in one slide. So the slide to your left describes the plastic industry. Today, the plastic industry is about 400 million tons in size. And it's 99.5% made out of fossil fuel. We are getting better and better in recycling. About 9% of that flow is made from recycled plastics. It's quite low, in my opinion. It's still very low. I think in about 30 years, we have tried to really harness the power of recycling. We have failed tremendously. And I think the first journey of that was when it was peak oil and oil was supposed to run out. It did not happen. So now we're on that topic again. I think what has also happened is this bio-based type of plastics. It's 2 million tons, made primarily in Brazil, or US. Not much is produced here in Europe. The expectations is for 2050 is that I love plastics, and it's growing. It will triple in volume by 2050. And this is believed still to be one of the largest challenges when transitioning from a fossil dependency to a bio-based society. There are no real scalable solutions. And bio-based is one of those that will absolutely grow. Recycled as well. The theoretical max is 60% will be recycled. I don't think anyone in the plastic industry believes in that, but that's the theoretical max. Then matching this, and this is coming back to Marcus Wallenberg, what you said in the early days, with where is the future? Where will the forest industry or the biomass industry grow? If we look at wood, I'm overestimating 180 million tons of lignin is processed every year in the wood and forest industry. What we miss, what we typically underestimate, is the amount of lignin that is actually used in the agricultural industry. This is the waste flows, the bagasse, the barley and other types of lignin that is annually produced and processed in the world. So the lignin is everywhere, is what I typically say. And typically in the history we have focused on this type of lignin source. However, in rice, in wheat, in corn and bagasse and soya beans, there are a lot of lignins available. And there's a lot of cellulose and hemicellulose as well in those raw materials. So this is what I'm telling Brussels. The raw materials, the biomass gap is not really existing. We have a lot of raw materials to compensate in this journey. So what we do in our company, we are buying lignin from forest or agricultural industry. I think, Hanna, you touched upon this. This is the one thing the forest industry needs to handle is the odor. And I'm not telling you that this should be odor-free, it's odor control. I brought some bags here as well so everyone can smell what it actually, a plastic smell would look like. And that comes from the craft process itself. So when you're processing this in terms of, from an agricultural way of processing biomass, you do not have the craft process, which actually generates a complete different smell on your plastic that is actually amusing or nice. What we do is we react in this in an epoxidized oil, a natural oil, and we generate a thermoplastic lignin that can melt and be shaped and solidified into a material that we call Renol. We then make it easier for the plastic industry. So what we're doing is we're bridging the forest or biomass industry with the plastic industry. So we're mixing this with a little bit of target polymer, which is polyethylene, polypropylene, or ABS. PE and PP are the two most commonly used plastics in the world. So they are everywhere. And then you take this thermoplastic material of lignin and some carrier polymer, and then you introduce that straight into the plastic industry and producing bags such as an e-commerce bags that I brought with you here. This has a white top layer without our material, and it could be brown inside or colored inside. And then, so this is also key of what we're doing. We don't want to change the infrastructure of anything. We simply want to make something that works within the plastic industry as such. No change in temperature, no change in processing volumes. The nice thing here is end of life. All the customers we are working with are obsessed with recyclability, which is a good thing. When we are designing our formulations, they are recycled in the open recycling industry. It's not a closed loop type of recycling, meaning that our products will be identified as PE if we mix with PE, and then sorted as PE and regranulated as PE and still functional as PE. Super important. So end of life, and recycling is working for us as well. moving on. So this is where we now are starting to take market shares. We are producing 2,000 tons outside Knivsta, Arlanda, in our factory. Primarily, we're growing here in the e-commerce bags, so the type of items you will get home when ordering online. The reason for that is that these brand owners are typically in this field using recycled plastics. A lot of those has gone over to paper bags due to a belief in sustainability when shifting from recycled plastics to craft paper. Issue they're having is that these bags are water sensitive, harder to print on, and are more volumetric, so logistics is more tricky. And also the gain in sustainability is challenged. The reason why we targeted this market, we want to get rid of the recycled plastic and start to introduce something that's more sustainable to recycled plastics. But what happens in the brand decisions is that the unit there is 40 grams for a plastic bag and it's 100 grams for a paper bag, meaning the paper bag is 2.5% or 2.5 times the plastic bag in price. So we're matching or slightly south of the craft paper, meaning price is super important. So we're cheaper than the paper bag. And that's why we're taking the early volumes from paper rather than plastic. I think that's something we could perhaps surrender in the short time in the paper industry. So the bags that we're doing, so this is our product, the granulates. Products that then is made is this e-commerce bag. Then we're doing, this is a cable hider that we're doing for the UK market. We're also doing Scania trucks. We're actually now taking off the box for making trucks out of our material. We're doing shower units now for the UK market. Just one shower unit in UK market is 200 tons. Just for the UK market, one shower unit. So once you enter the market, each item is quite large in volume. And this is PP. This is our strongest candidate. We mix in up to 40% of our material, preserving material properties, and then you can have a range of different colors. This is in the piping and construction industry that we're now launching as well. This is just giving some love back to Mr. Wood. You said it yourself, and Mr. John Ralph. The complexity of lignin. So we have worked with 35 different lignins. I think this is 25 different lignins. And now I have rated them, so different source, different processing conditions. So we have built up a lot of knowledge on lignin, and we've been told about this, my quality of lignin is the best, and so forth. So when we are doing this, we arrange it so a standard ABS plastics is about 17.5 in impact strength. So the impact strength is in the y-axis. We end up with a variety. So this is mixing the same stuff, about 10% of modified lignin in ABS. We have the range from 5, really, really deteriorating the ABS quality, or we have 25. So doing the same stuff, but with different lignins, generate very different results. And in order for us to understand this, we need to go back to the structure and the miscibility and the fundamentals of what lignin is. So when we are, this is just in order for me to complicate this even further for us. So when we look at extraction methods and trying to understand, is the process method, is it the raw material, is the quality of lignin, what is important? What we have been told before reading your stuff, we have been told that Organosolv is the purest and best lignin you could ever buy. And before reading what you've done and the importance of the composition of lignin, the characterization, what's it built up of, one would have thought that in an ABS that the purest lignin will gender the best results. This is the worst lignin, lignosulfonates. It's about 30% of ash content. And we generate typically the highest performance in ABS. We have done this. This is only ABS. We've done it with polyolefins as well. The same type of fundamental study on where do we buy the lignin and what influences this. So coming back to miscibility in an ABS system or miscibility in a polypropylene mixtures, it comes from the chemical structure of lignin rather than the amount of ash that it contains. So the industry start to perhaps design for fit for purpose lignin is what I'm trying to say. Just touching base on what lignin, it says Renault there in the headline. It could say lignin as well. So utilizing the entire tree, this is where I come from. This is where I believe the oil refinery has done beautifully. I think we could do the same in the biomass industry. Drop-in, this one thing I would like to highlight. I've already highlighted it, but what we are trying to do with lignin is basically to feed the oil and gas and plastic industry with a different raw material. Instead of feeding it with oil, start feeding it with lignin and then go on and do your product. And with that, I would like to thank you.
Thank you, Christopher. What a success story. Do we have any questions for Christopher? Up there? No? So what do the plastic industry think about you?
The ones that believe, let's start with the ones that doesn't believe in us. They say it's the wrong color and it smells like wood. It doesn't smell like plastic. So those are the ones we typically walk away from. The other part of the plastic industry, what they truly like is that they don't need to change anything. We're running their plastics at 200 degrees Celsius with some kind of or throughput of X, and they're still using the same throughput, the same temperatures, and you're actually making the surprise that it actually works from the very start. So ease of implementations is what they're really happy about.
Thank you. Anyone who has changed his mind?
So
I have a microphone here.
The polyethylene and polypropylene market is about 200 million tons. ABS about 10 million tons. And then we are targeting the colored side of that. So 50-60% is colored, and the rest is white or transparent. We don't do white and transparent. So about 100 million tons is the market.
Okay. So thank you very much, Christopher.
Come and smell.
Yeah, so you have your test samples with you, so go and see Christopher later. Okay, so we are continuing in our program, and if you have had an early program, we have a small change today, and now I would like to invite Malin Alpsten. And Malin Alpsten is working at Bright Day Graphene, and she will learn us more about high-performing batteries based on with lignin-based graphene. So Malin, please come up.
Thank you. Thank you for inviting me. Yes, my name is Malin Alpsten, and I am the co-founder and CEO of Bright Day Graphene. And today I will tell you about the super material of the future that has been awarded the Nobel Prize, that has a higher market value than gold, that can improve the performance in a wide range of products, and that we make from lignin. And the material I'm talking about is, of course, graphene. This fantastic carbon material that most of you might have heard of, that is 200 times stronger than steel. It's flexible, transparent. It's a superconductor. It conducts both heat and electricity with no resistance at all. And it's also a 2D material, only one layers of atoms, which means that we get a lot of surface area, but almost no weight. And all of these properties makes graphene the perfect candidate to revolutionize a wide range of products and markets. But the discovery of graphene got a recognition by the Nobel Prize in 2010. So it's been 14 years now, and a lot of the graphene potential has not yet been realized. And this is because it turned out to be quite hard to mass produce graphene with all these fantastic properties still in place. So there are two main ways to produce graphene today. One is the CVD graphene, where you produce graphene on a substrate, one layer. Here you get a lot of the properties still there, but it's made on a substrate and kind of hard to transfer without lowering the quality, because it gets injured. And the other method is the graphene nanoplatelets, where you take graphite and exfoliate it mechanically or electrochemically into a graphene powder. You get nano-sized flakes that are often a bit thicker, not a one atom layer. And this means that you lose a lot of the properties that is so fantastic about graphene, especially the electrical properties. So what we wanted to do was to make another kind of graphene. It's still a graphene powder, but with larger and thinner flakes, which gives it more of the electrical properties. So we developed this process that we patented, where we transform lignin into a graphene powder that we call grapholium, that has the large thin flakes that gives us better conductivity and makes it suitable for the high-tech applications, like energy storage and electronics. And this is a high value-added process. Graphene is adding a lot of value to these products. And when it comes to the lignin that we use, we developed this process based on craft lignin, actually from the lignobose process that Hannah was talking about. And we have mostly used the softwood lignin that is mostly available here in Sweden. And we have tried a lot of different lignins, and what we have discovered is that we are not that sensitive to the method of producing the graphene, anything, or the lignin. They all work for us. We have quite a robust process, and it's not so sensitive. But the type of woods is makes more of an impact on the material. So hardwood works in general. We've tried eucalyptus, and that makes a beautiful graphene. The properties are slightly different, so it's suitable for different applications, but it's a good material. But we also tried birch, and that does not work at all. Neither does grass lignin, unfortunately. But otherwise, most can be used, and we prefer the low molecular weight lignin. So if we can somehow modify the genes to make it more low molecular, that would suit us very well. And the material that we get is actually a reduced graphene oxide with an oxygen content between 3 and 10%. And the most important feature for us with our graphene is that we call it green. because it's from a renewable source instead of a finite resource that most graphene comes from graphite. And graphite is also, since 2020, on the EU critical list of critical raw materials, because we use a lot of graphite in Europe today, and we need to import 95% of it, mostly from China. So to be a more resilient society, it's good if we can move away a little bit from graphite or find other sources to make it. And then we have these large thin flakes that we get that gives us a lot of good properties. And another good thing about the way we produce it from lignin is that we get a more controlled process. When you produce from graphite, the quality can differ from batch to batch, depending on the quality of the graphite. You also get a wide variety of the size of the flakes when you make it from graphite, while here we get a much smaller variety. So the reproducibility of the process is a lot better when you make it from lignin than from graphite. If you look at the graphene market, I initially said here that graphene has a higher market value than gold. And this is both true and false, depending on the quality. Because you can find graphene on the market for as low as $20 a kilo, but it's also sold at the price tag of $100,000 a kilo. So there's a big variety. And the most expensive kinds is the CVD graphene that is perfect quality, but that's almost only used for research, because you can't really base commercial products on a material that cannot be mass-produced and is that expensive. But a lot of what is sold as graphene today is not really graphene, because graphene is one layer atoms, and we say that between 1 to 10 layers, it still counts as graphene. But a lot of what is sold, and especially down here with a low price tag, is actually more of a nanographite. It can be 50, 100, or even 300 layers sometimes. But for some applications, this does not really matter. One application that we have seen a lot is in sports equipment. Head has had a line of products, I know tennis rackets and alpine skis, for instance, with graphene in them. And for that type of application where you don't need the conductivity of the material, it doesn't really matter. Same goes for a lot of building materials. At the moment, there's a lot of focus on mixing a small amount of graphene into concrete, and not because it makes it stronger or more flexible, that might also be a feature, but because it lowers the CO2 emissions by 20%. And that is a huge market, of course, although the price tag is a bit lower. There are so many different applications you can do with graphene. That is the blessing and the curse of working with it. The same goes for lignin, I think. And I'm sure that we will, in 20 years or so, have graphene in a lot of products around us. But when you go up this line, you need more and more of the conductivity in the material and the surface area sometimes as well. And for our materials, since we have the larger, bigger flakes, which we define as a higher quality, we have chosen to focus a lot on the energy storage market. So we try this in batteries, in supercapacitors, and in heat sinks and some other things. Material support sometimes, because there are companies making bio-based materials that want to have a stronger material, and then they come to us to have the bio-based graphene to be able to still stay 100% bio-based. But we made trials in batteries, and what we learned is that we can prolong the lifetime of a battery by 2.8 times when we mix in a very small amount of our bio-based graphene. And it's probably more than 2.8 times, but we couldn't, the project time was limited, so we had to stop there. But that's one feature that we get. But we also know that we get a faster charging of the battery as well, enables that. But also, we see that the fire safety of the battery gets better. We haven't done this test, but I know that another company has, where they took one battery with graphene in it and one battery without graphene. This is lithium-ion batteries. And then they took a gun and shot at them. And the one without graphene exploded right away, while the lithium-ion battery with graphene in it just got a hole through it where the bullet went through, and nothing else happened. And I think this area is very interesting because already there are ferries or garages where you're not allowed to have your electrical car. And I think there might come a lot of rules and regulations around this eventually, where the battery manufacturers will have to find ways to make the batteries safer. And then we can be an enabler for this. And I'm not doing this alone. I have my colleague, Anna Karlsson, who is not here today, but she is the brilliant inventor behind this idea. And we are currently at a small scale still, but we are getting requests for larger samples than we can provide. So we need to scale our business, and that's the plan to do in the upcoming years. We plan to build a plant in the northern part of Sweden, where we can produce a ton of graphene material a year from lignin. So that's all I had. Thank you for listening.
Thank you very much, Malin. Very exciting. You can see there are many different applications of lignin. Do we have a short question for Malin? Yes, please, Monica.
Thank you, Malin. Very inspiring talk. And Antti Asikainen already touched upon our report, and this was one of the killer applications that we saw has the potential for really added value materials from lignin. Are you willing to say anything about the challenges that you still have to get to the a lot of applications, or what are the main bottlenecks still?
For us, it's the bottleneck that we are facing now is that we can produce so little material, so we can't explore in the speed that we would like to, and different applications. So that's definitely one. But otherwise, it's also that the graphene market. Graphene is a different material to handle with the 2D material, so we need to build knowledge, and it's getting better and better.
Okay, thank you.
So, thank you very much.
Thanks.
So, we have already come to our last presentation in this MVP seminar today, and I would like to invite Britta Nilsson from Stora Enso, and she will talk about how bringing nature's binder back to wood. So, welcome, Britta.
Thank you, Monica. I'm thrilled to be here as the last presenter. And as we've delved into the fascinating world of lignin today and listened to these very interesting topics about this material, so now let us look at yet another innovative application, industrial application where lignin can be used, and maybe the most obvious one as well, because now we take lignin, the nature's binder, and use it exactly as what it is, a biobinder in making wood products. I'm Britta Nilsson, and I'm responsible for sales and business development of our lignin applications at Stora Enso. So our vision at Stora Enso is simple, yet very powerful. Everything that's made from fossil-based materials today can be made from a tree tomorrow. Currently, about 90% of all the materials and chemicals are fossil-based. At Stora Enso, we want to be at the forefront of the industry and change these statistics. And that is why we have developed Neoligno Biobinder. So binders are crucial components in many of the materials that we encounter today. They are present in our homes, in the walls, in the floors, in the furnitures, everywhere. And traditional binders in these materials have more than 50 years of history that are based on fossil-based technology. And not only they are fossil-based, they are also containing chemicals like formaldehyde and isocyanate that are hazardous both for the people and for the planet. So Neoligno Biobinder is bio-based and it doesn't contain any harmful chemicals that may emit from the end product. To better understand the consumers and end users' knowledge and awareness about the emissions from the materials that are surrounding us, we conducted an end user study back in 2022 in Germany and in Sweden. And the results from this end user study were eye-opening. We found out that 88% of the consumers, both in Germany and in Sweden, they are aware of the importance of their indoor air quality for their overall well-being. And 50% of the consumers are also aware of the consequences of poor air quality and how it affects their health in a negative way. And this is so important for them that majority of the consumers are willing to pay more for emission-free products. However, the knowledge about these hazardous chemicals like formaldehyde and isocyanate is quite low among consumers. And here it's very important that the industries and legislations take the leading role and set clear guidelines and restrictions and find better options. So Neoligno is a better alternative to traditional binders because it doesn't contain any harmful chemicals that may emit from the end product, and it is more sustainable. So it is more sustainable because the biogenic carbon in Neoligno is circular in the atmosphere. So we no longer take carbon from the ground, put it in a material, and then release it to atmosphere at the end of its lifetime. And by reducing our reliance on fossil-based materials, we can lower our carbon footprint and also grow a more sustainable future. So let's take a closer look at the carbon footprint of Neoligno. And please note that this information is currently under third-party validation. But just to show you the first initial result from the LCA study, you can see that the biogenic part of this graph, the green part, is slightly higher than the carbon-based part. And this can be interpreted as carbon neutral. So by reducing our reliance on the fossil-based materials, we can lower our carbon footprint. And the biogenic carbon is circular by nature in the carbon cycle. So you may ask, is Neoligno available commercially? And I'm pleased to announce that yes, Neoligno is commercially available in industrial scale. It is ideal to be used in mineral wool insulation and wood panels. So mineral wool insulations can be used in floors, in the walls, in the ceiling, roof, and also in industrial environment, in pipes, etc. Wood panels, there are various different type of panels. That is what I learned when I started working with Neoligno. So particle board is one of them. It's the simplest one. And there are many different type of particle boards, and that is the part that you don't see in your furnitures. And then we have plywood, formed plywood, and that is the furnitures that you can see. For instance, the formed legs and tables or chairs. And we have customers who have already launched product based on Neoligno. I will later on tell you about our first referenceable customer. We have, for instance, IKEA, who have tested this in their formed fiber chair with good results as well. But these are still the first early adapters in the market, and we hope that rest of the industry will also follow suit and implement Neoligno in their products. So Neoligno is Stora Enso proprietary, and it is in liquid form, and it's not only one product, it is many different formulations. My colleagues within R&D, or my very clever colleagues in R&D, they may be listening online, they have formulated many different Neoligno formulations adapted and tailored for the end product. So there is Neoligno that is optimized to be used in particle board, Neoligno that is optimized to be in mineral wool, plywood, MDF, OSB, etc. It doesn't contain any harmful chemicals like formaldehyde and isocyanate, so it ensures healthier indoor air quality and also working environment. And with Neoligno, our customers can offer their customers a bio-based solution and lower their carbon footprints. So the Zero panel was the first commercial product where Neoligno was utilized. And this panel was commercialized and launched in 2022 by one of the forerunners in the industry, the Finnish company called Koskisen. And as I said, we have several customers who have tested and have already either launched product or are in the process of launching products with Neoligno or new product developments. But still, they are the forerunners, the early adapters, and we hope to see several following the trend. So, by that, to conclude, Neoligno is beneficial both for the planet and for the people. For the planet, because we can lower our carbon footprint and reduce our reliance on fossil-based materials, and for the people, because it doesn't contain any harmful materials that can emit. So thank you for your attention. I hope that you are as excited as we are about Neoligno and its potential.
Thank you, Bitta, for this interesting presentation of how to produce big volumes based on lignin. Do we have any questions from the audience to Bitta? What has been the challenge for you?
The challenge for us at Neoligno, I think it is to have the customer, to find the pioneers that want to implement it. This is a fantastic product, and it is plug and play. We say that it doesn't need any big changes in the industrial trials, but it is to come from the industrial trial to an actual product. And I think it is also following the market, because it is right now it's not so many renovations and buying furnitures, etc. So I think we will see the trend turning when we have the better economy as well, when people start renovating and buying furnitures.
We're looking forward to that.
Thank you.
Thank you very much, Britta.
Thank you.
Okay, we have already come to an end of our 2024 Marcus Wallenberg Prize seminar. We know that United Nations has these sustainability goals, and we have to try to find solutions for them. And I think we who are here, most of us here in the room today and also online, are working with wood or bio-based resources. So I think this is the green gold for the future. We have been working with wood, and wood has been used for hundreds, millions years as construction, fuel, but we have also tried to make new materials, chemicals, and new properties. And since we have this lignin, we are using the cellulose mainly from the wood, but we have for a long time have this lignin from the pulping processes, and there has been research to try to give this lignin added value. It has primarily been used as an energy source, but since we have these challenges now, it has been an increased search for using this for more high value added applications. And as we started up this morning with Claire, she said that you can do anything from lignin. Yes, we have seen many examples of that, but we also need to have the structure, properties, and need these advanced techniques to characterize this complex material. So we were listening to our laureates this morning, and Professor Boerjan, he talked about how to improve the wood quality from the system biology in Arabidopsis to application in trees. How we can modify lignin already in the plants to give higher volume of the material we would like to have, and increase the yields. And then we listened to Professor John Ralph, co-laureate today, and his amazing work about organic synthesis and how he has used this advanced analytics to understand lignin biosynthesis, structure and processing. And we saw a lot of this NMR spectra and how this has been used to characterize the lignin. And I think it's amazing how this work has progressed. We heard that Professor Boerjan and Professor Ralph met at the conference many years ago, and networking, mainly talking about what they are doing, and this was the found for this very long cooperation and successful cooperation about lignin. So this has been a solid ground for this after break presentation about innovations and startups, and we started to listen to Monica Österberg, how she is using lignin nanoparticles for various type of application in coatings and emulsions, and how important it is to know the morphology and chemistry of the lignins. And then we have Hanna from Valmet describing that we already have lignin. There is lignin in large volume, and we can also have it with the properties we would like to have. It can be super pure without ash or metals. It can be odor-free. So there are many potential openings for use lignin in novel products. Then we listen to a very exciting presentation from Remy Buser and Bloom, and how we can make novel materials from lignin and also other agricultural raw materials. And you have already proceeded in your upscaling, so that will be very interesting to follow in the future. Then we have a couple of examples of our startup companies. There are many out there now. It's a very big interest to do things with lignin, but we have an example from Christopher Carrick and Lignin Industries, how we can substitute oil in plastic materials, instead use his special technique for modifying lignin and have it to increase the bio-based material in plastic applications. And then we have Malin talking about this lignin for graphene, and how it can be used for making safer batteries, and there are a lot of potential application if for many of these things we are doing, we need to be able to go from the lab and scale to larger volumes. As we have heard, there are demand out there who want to try our materials, so we have a lot of things still to do. And finally, we listen to Bitta about this bio-based neolignin, who can be used for making large volume of construction material and help them to reduce the carbon footprint and also this carbon dioxide emissions. So a fantastic morning with a lot of inspiring presentations. And we also have this meeting with our young professionals. We have had the opportunity to see what 30 young professionals are doing in their ongoing research work. And you are representing many universities, institutes around, both in Sweden, Finland, Europe, and abroad. We have contribution from Brazil also. And the foundation is very happy to see that you are here, because that can make us, all the seniors here, to feel a little bit safe for the future, that we will have many new products and processes based on our renewable material. So today we have had great networking, I would say. I hope you have built your valuable novel contacts and also connections for future collaboration. So finally, I would like to thank Marcus Wallenberg Foundation, and also thank you the laureates, Professor Boerjan, Professor Ralph, and all the presenters who have presented their research here today, and seniors, young professionals, and the audience, both here in Grand Hotel, but also for all of you who are listening and following this meeting online. So if you want to listen to something again, the presentations will be available later. So thank you very much.